Hello everybody, welcome back to the Genome biology class. In continuation of the previous class, we're going to study replication of DNA. Among previous class, the central dogma the highlight at the PVT replication of DNA, transcription, translation, the highlight details replication of DNA. Replication of DNA habadi the DNA DNA replication of DNA. The synthesis of DNA is done as replication. So how is the DNA synthesized? Synthesized we know DNA by the method of semi-conjugative. Semi-conjugative and high semi-conjugative. See my propose to Watson and Crick. Watson and Crick have the partners proposed to give them. 1953. According to it, the two strands would separate and act as a template for the synthesis of new complementary strands. Semi-conservative habit DNA strand because the structure of DNA strand since act as a template strand template template and this also is a template for the synthesis of new complementary strand. Complementary strand complement of a strand after replication, each DNA molecule would have one parental and one newly synthesized strand. Details of the mechanical. So the DNA is strand. DNA is strand. Aduga two strands to me. Two strands to me. I look at the segi sina parental DNA. The parental DNA say parental DNA name. Parental DNA sa that's minor. Katalaga segi Tommy do it and the manus finance had to pay type of Tulakani, Aduka do the each strand the gay. They don't get each strand the gay minor. Oh, Simakadori as mine. But I did DNA Madaka Sungani, DNA Anilani. So DNA Madaka DNA Anilakose Aduki replication process in the to the shot. Other process called semi-conservative nature, semi-conservative. And a cow have got. I do command of the to see tempted and sin any. Must it have a parent strand or as in blues in a parent. Have parent strand. Sinner, newly synthesized strand. Or you can also an original template and higher CN. For seeing experimental proofs, how the semi-conservative knee. For semi-conservative in experimental proof to say, can experiment to give no Hamilton, the Michelson and Star. Michelson and Star have a gay, by one or full name them Matthew Michelson and Franklin Star have a national experimental kaka, semi-conservative native proof to give. Hammer the Chanting and counting at the class. Counting at that, each of them say DNA and the any science to win science to win conservative kind of the book. Proof can proof to give Hamilton, Michelson and Star have a sinner. Kaineva semi-conservative nature, so the proof to give. Other command proof to 1950, the proof of the man get the E. coli have a bacteria. E. coli have bacteria is low left. And the mass in a correct the growth of the medium and the growth of curry medium. Ammonium chloride. See, ammonium chloride in the chemistry, the 4 is mining at them. Ammonium chloride. 15 ammonium chloride. Have the radioactive element now. 15 ammonium chloride. That is 15N is the heavy isotope of nitrogen. That is the heavy heavy nitrogen isotope of nitrogen. This is the growth of the as the nitrogen source. This is the incorporation of 15N into newly synthesized DNA as well as other nitrogen containing compounds. Combine to get the E. coli. E. coli is kind of growth of Hamilton that's Kariba 15 have a high isotope nitrogen have a pochi nitrogen have elements. My isotope only lay in 14 have a N14 have a my mass number 14 neba ma mana mass number 15 neba ma isotope ata na chemistry tamilni isotope ata di same atomic number different atomic mass ko nitrogen je n14 neba le n15 neba le siu ma chu kai kawi isotope adu isobar ha ga ni chemistry ta isobar ha ga na mass number na manaraga atomic number na khena ko adu isotope si isotope nitrogen ni isotope si da asagi E. coli si grow tawone isotope si grow tawre and was grown for many generations in a medium containing ammonium chloride si grow tawre si grow tawre kanda kai do ge da asagi nitrogen si masagi manas grow tawre ke si na si nitrogen ji resource and ba habadi DNA replication ta odama gi source to kai de lo to no nitrogen source to kai de lo to no si ammonium chloride si source lo to ne and this result in incorporation of 15 and into newly synthesized DNA as well as the other nitrogen containing compounds so then combine together aduna DNA synthesize to do ne ismaina habadi magi strand animal si habadi DNA do ka de grow to no 15 and ammonium chloride the growth of which have nitrogen isotope N15 ji yawo ko sida growth of matam sida the replication ja tawde DNA synthesize tawde DNA synthesize tawde ada the both the strands to the ko de growth of ni both the strands to ka dorge sai strand animal sa 15 and oil have strand animal si su ka yawo 15 isotope 15 and yawo si su 15 and yawe then mathang sida kedoge da here ko centrifugation ata the centrifuge to body kada unthala ni ko unthala kan da separate to ni cesium part da hapaga separate to ni aduna si separate to si ka gido na sa si heavy DNA or heavy DNA molecule sa ka de hanigi normal DNA to hade isotope yaw da ba normal DNA separate to gibane heavy DNA do na ka de le hoba heavy DNA na makha da batam da le ni sina batam da le sina ko heavy DNA batam da le na heavy ata ade ka do ni makha da dum da le ni heavy ha ba na centrifuge to matam da centrifugation na chemistry ta centrifugation ta di kan na unthala kan da ko high speed to kan da lactate particles na top the layer ni heavy particles na bottom the layer ni ha ko ada ka do ge da si the cesium price da centrifuge to matam da sagi si makha da kai phangi matam si heavy DNA phangi ko heavy DNA gi start je phang ada ka do ge heavy DNA start je phang matam da ka do ge da si lo ko ado ayang ma su to lok thok ke le ayang ma su to lok thok the then next the cells were then transferred into a medium with normal 14N. Had the 14N in a light over cop. Maggie can say, she's a light over medium to happy. Light medium over medium to have that day combined together. She'd have them to the Asagi, being what kind of in the light medium that will grow over kind of say, parents stand the Kai order. Parents stand the Hanagi DNA log whole government. Should he have DNA? She had 15 ammonium chloride 15 and the growth of DNA. She looked whole kind of go to Hodina. She can also 14 and she don't grow to Hodina. She get 14 and still growth of what Tamsida Kadogi the Asaki parent stance. Parent stand to Kai wedding to thee. But the 15 and the growth of one parent stance do. Parents stand to Kai where this is 15 and. Experience stand 15N oil. 15N oil. Aduga newly synthesized strand the kai oil again because it is grown in N14 the growth of unina newly synthesized strand the kai oil again N14 oil. Yari ko. The might of the Sigumba, I said, I'm not getting one Siguma meow, phone of the knee, so I can then 15 oil bar in heavy with Amadana. So I told me I'm a thing to do never. Thing didn't go. She wrote it. Oh, the she now. She like I don't want to see the replication to see the Muka do get up. On season of the money, she look at together as a replication to continue grow on see the qua. Sagi N14 that don't grow out. N14 that don't grow out. She Kari from air. Kari from air have at them that's intermediate have the she porcher. Hung air. She had a tongue air, I don't know. She don't continue growth on Banana. 시주 방해. Two types of DNA template 방해. 방해권. Two types of DNA 방해. 방해간 그게 form 또 N14, 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 N14 누가 last hybrid ama yawi. Series by DNA parent stand kay dung orani, sidung orani. Parent anu le sinisa stand du sai sida grow torabani na dum sidung phangya. Adi sidu N15 sida sidu grow dum torabani na N15 orjata sidu mai parent stand na kay orani. Parent stand na si blue color soire. Si parent stand orya. Adi new le sinisa stand na khapa ani. Adi sina parent stand kay machujo orani pink color si orani N15 je aduka jagi dosina. N14 je. Adan takpode sagi sina tai si game te sagi parent stand adum omam magi parent stand kai orani sa sigi parent stand di ado takpode sigi pink je ama ton de yaw go poche sida su yaw tra sida yaw to de takpode sigi poche adum parent stand ama oina adum yaw e habni da pada parent stand de conject to go leijana ada semi conservative model na ko semi conservative model na ko wa. After one generation of transfer, that is after 20 minutes, E. coli divides in 20 minutes from 15N to 14N medium. The DNA that was extracted from the culture was found to have a hybrid or intermediate density. After another generation, that is after 40 minutes, it was found that the DNA extracted from the culture was composed of equal amounts of this hybrid DNA and of light DNA. Then the last day kay phange hybrid DNA siga light DNA is equal amount of phange. Then the takpadi the DNA replication is semi-conservative anatai. In 1958, Taylor and colleagues conducted similar experiments on Visha fava means by bin sabanikor. By using radioactive thymidine to detect distributed newly synthesized DNA in the chromosome. Sige Taylor have a mission amu kadukida on a radioactive element sagi semi-conservative oiba simu check to man. Adasu kay phange semi-conservative oide se have the parent stand template oina ek tawi aduna 50-50 phangani ba parent stand yawa amada parent stand down 50-50 phangera. And using radioactive thymidine to the distribution of new DNA, they also concluded that DNA in chromosome replicates semi-conservatively. Muna su kay hydra semi-conservative ta oina replicate ta oisi muna su hydra. Then DNA replication se combined with DNA replication se combined with DNA replication is a highly enzyme dependent process. DNA replicate ta DNA se combined various enzymes ma'am changi adagidi newly stand hapakuma tamda karine hapakadoino kaina sagi DNA stand sigai doino adugi enzyme various enzyme changi ba sigi enzyme loine na kadoni and there are many enzymes involved in DNA replications which includes enzymes maru in harabada DNA dependent DNA polymerase then helicase then ligase etc. Hamagi DNA dependent DNA polymerase important ba enzyme. Masina kay the wheel. It helps in the polymerization catalyze and regulate the whole process of DNA replication with the support of one other enzymes, deoxyribonucleoside triphosphate. Or deoxyribonucleoside triphosphate are a substrate as well as the energy provider for the replication process. DNA polymerase is of three types. Prokaryotes ka eukaryotes din sa DNA replication ni magi enzymes yung kailangan ko. At prokaryotes di magi isam na DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. Then prokaryotes like bacteria gumba E. coli da replication tong DNA polymerase ya. DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. Whereas in eukaryotes di for example, in human beings the eukaryotes today, karamba DNA polymerase nahi hamatada alpha, beta, gamma, delta, epsilon. Then, DNA polymerase 1 DNA polymerase 1, it is a DNA repair enzyme it is involved in 3 activities. My DNA polymerase na tawag kasi sa DNA strands hapsalagtuni naman. DNA strands hapsalagtuni naman. Sino ko? DNA polymerase, DNA replication to bagi process. DNA replication na kay ni ba? DNA strands si madagi suppose DNA replication di son na Pachu ani sino kang na ba? Sino ba? Sino kayo ni? Parent kino ne ba ko? Parent star ne ba? Ada sino kakala ni ko? Kakata di ba? Sumaina. Sumaina yan niya. Kakalin kalo. Sali adi then newly synthesized strand. Newly synthesized strand is mine. Have to look for them. Or the song section have to look to them. This is a newly synthesized strand. Newly synthesized strand, at the start is same you got. Then after that, also DNA is formed only. Smile. Sini ko. DNA polymerase 1. DNA polymerase 1. It is a DNA repair enzyme. It is involved in the activities. Okay, the 5' to 3'. 5' to 3' polymerase activity. 5' to 3' polymerase activity. Then 5' to 3' exonuclease activity. Ay makal lalo. Sino yung tawag kinigo? 3' to 5' exonuclease activity. Then polymerase 2, silang kadawin, it is responsible for the primary extension and proofreading. Polymerase 3, silang kadawa, it is responsible in vivo DNA replication. Number 2, the helicase. Helicase, helicase is the enzyme that unzips the DNA strand by breaking the hydrogen bonds between them. Thus, it helps in the formation of replication fork. So, you know the body helicase DNA strand of DNA strand of DNA strand DNA strand mark to sige lagi purine nga pa si mark the bond kay bond na si mark the labor bond edin na maga si mark the labor si sida nitrogenous space siya ni na nitrogenous space edinin nga kanaga space to diba edinin ka bond form to diba edinin ka kanaga bond form to ni kanin ka Lagi cytosine nga. Samin nga. Triple bond. See, marak si the bond. See, this is the hydrogen bond na siya. This is a hydrogen bond yan. Sige, hydrogen bond siya. Sa hydrogen bond siya, kaktuin niya. Sige, hydrogen bond siya, kaktuin niya. Kanana kakani, sige, hydrogen bond siya, marak sa minus 6 kaktuin niya. At yung tawag, kakpoy tato ko. Kaktuin niya. Sige, kakpoy kanana, kakaino. Ay matagda. By the enzyme, helicase have an enzyme. So, helicase of the enzyme is unzips. Unzips, I tell you, chain the one thing double to change the cut, ching double, unzip double. Chain down, make it change the ching cut, ching double. Unzips double, ching have the CCI. By breaking the hydrogen bonds between them, thus, it has the formation of replication fork. Ado kay form to ni. Replication fork, replication fork, the DNA replication to the helicase na swile mapochi DNA strand si schedule ni sida unzip to ro then as it sign to ro si sign sigaro address mana this is called replication fork ko have the Y shape maleda replication fork replication fork then topoisomerase and this enzyme release the tension in the DNA strands created by the unwinding of DNA. Unwinding of DNA si unwinding helicase ito kali ko. Pero matuno di sana unwind to sida matak dari tension na mag-create ako ni ba. Sige tension na mag-create ako siya. si tension nama create tawbo si kanana release tawon gi ha matamda topo isomerase sina release tawdone topo isomerase release tawni then mathangda ligase habi enzyme dina kadawni in which the Okazaki fragments habdi sida fragment macha semakani sona kan side sida ko si matat khar yauri matat sambasina kanana samdoino sida ko side matat semakani fragment macha adu si fragment dai sia kanana muk samjan bidoino hai matabda DNA ligase na samjan dai ne DNA ligase na samjan ne then that endonuclease sabi sina primase hana na RNA primary mo intolani then endonuclease ay matfana hara magi the replication process involves the following steps bata karino initiation or formation of replication fork replication fork the form theory the replication starts at a point called origin or origin na pasi kaya paano origin na pala replication to wamja sida sini ko replication fork kasi say unwind to ya with the unwind to ko kari enzyme unwind to ba helicase na sa chato matamna ka daw ni sila unwind to gani so na ko sini ko helicase ay then kado ni ha matamda sige DNA polymerase laani ko DNA polymerase si ORI region jingo DNA replication sai digi hauri na khala ada si region di demo ORI region is origin DNA replication to guma pase haguma pase origin a unit of replication in one origin is called a replicon. During replication the two strands unwind and is separated by breaking hydrogen bonds in presence of an enzyme called helicase have enzymes in a break tawari. Unwinding of the DNA molecule at a point forms a Y-shaped structure called the replication fork. See the Y-shaped structure called replication fork. Form tawari. Y-shape order ne sina sai chakani. Sigi part ne man Y-shape si chak to sumai. Si part ne Y-shape. Then mathangda elongation and polymerase. Sagi kakthak palwara kanda DNA strands newly synthesized to kadoy name ko. Shikanan si the polymerization hai din. The separated strands act as templates for the synthesis of new strands. The DNA replicates in the 5' to 3' direction. See, very important. Kari direction the standard DNA standard 5' N naman like 3' so na kanina 5' N already sige N si kayo ni 3' N so na kanina 3' N wala ba som side sina 5' N ha kana hala DNA replication ni karon gi hauge matamda 5' to 3' in direction habidi 5' de sile 5' to 3' in direction is my to ne 5' to 3' direction is my to ne replication. Then deoxyribonucleoside triphosphate is a substrate in egg and provide energy for polymerization. See the DNA polymerase because this is a DNA polymerase. DNA polymerase is energy polymer having synthesized to newly synthesized to polymerase have enzyme to hold it. Ada makala DNA adenosine triphosphate G sina kay hayamani G sina deoxyribonucleoguanosine hayamani ko deoxyguanosine triphosphate hayamani sina deoxycytidine triphosphate hayamani deoxy kay thymidine triphosphate hayamani sige sina mai ni base kano kai do nitrogen base kai do loina pura la ani DNA ke strand ka yogi nitrogen base yaw do nitrogen base na kai do gi do loina pura ga la ani ba si DNA polymerase. Firstly, a small RNA primer is synthesized in the presence of an enzyme primase. Aduga howdringayda ko DNA polymerase na thabak tawdringaysida RNA primer ama changi. RNA primer ama changi ko. DNA polymerase na thabak tawdringaysida RNA primer ama changi. RNA primer sina kadawi sida RNA primer sigi red idisa to the red to this is a RNA primer to the red cha RNA primer ne. RNA primer is later the DNA polymerase law. RNA primer is nafaganda. Macam tani ko singles macam tani ko si macam katani. Si mal nafaganda ka dulu ba. Masih lah aganda then DNA polymerase. Jadi jangan jadi. Sung dulu ni. Synthesize dulu ni. Ada DNA polymerase na laki hai tadi it requires RNA primer. Should I say? For a small RNA primer is synthesized in the presence of an enzyme. Kalau kena ni enzyme, RNA primer na sumber RNA primer sumber ni kena be enzyme no, primase sabe enzyme ni, sumber ni, primase sabe enzyme ni sumber ni. Then in the presence of an enzyme, DNA dependent DNA polymerase, many nucleotides join with one another to form primer strand and form a polynucleotide. Then DNA polymerase lani then sinap talaga. Magi chana ba complementary strand with sumba orani thana suppose na kanda adenine laita adinang sidamai complementary strand kai orani complementary strand oita adisona kanda guanine yawo pochilani sida T oita di sidana kai porani C porani A oita di G porani by the complement strand sinja zer hatulani by the DNA polymerase. And polynucleotide chain is formed. Then polynucleotide chain is found from the RNA. Endonuclease is single strand or double strand cut in DNA molecules. Endonuclease hai tanding marak takak kadab DNA strand mamatay dakak mo tama manung damay marak takak mo sina endonuclease. Then single strand binding proteins. Single strand binding protein is short form the SSB na yawani ko SSB. SSB it binds to a single stranded DNA and protects in the forming secondary structure. If DNA is the cold to sandi ba the secondary structure form to the secondary structure form to prevent it is prevented by the single stranded binding protein. Single stranded binding protein sina kanato video. Protecto video. Then sini ko. Siko na lagi. Ada strand naman sina kumain to kita. During one strand is formed in the continuous straight 5' to 3' direction, this strand is called leading strand. Aduga sona kani side sila di kedong kita. Sila dong sona kani kena ating mambilay rebels. Kaya gino na sige primer sina somda napakalay. Aduga sige DNA polymerase jakidonium. Aduga kadang sila di adum malagta there is no fragments. Aduga kadang ni ham kadang fragment na itabida. This is a leading strand. Leading strand application is small this is a leading strand called. Then the matang dana deoxyribonucleotides it is a substrate here. Firstly, a small is in the presence of an enzyme primase. The other strand is formed in small straits. So na kani sites nila okaja ki fragment ko rebo ko si part sine. Sida madi RNA primer single changan ko masina ko maito gida kayo si same direction is fine the kinam neba. Sida fragment macha in the form to it have the amana som the chatpa do sagi same direction da sagi kana chat kinam neba. Si sina RNA primer is side 5' to 3' na sung yaba tro non som ni manage som this minus som na synthesis to guru ni amma sina som na minus synthesis to ni kada sina RNA primer to sidale RNA primer sin dapsula ni RNA primer la adi kana la ani DNA polymerase la ani RNA primer sidale RNA primer la adi DNA primer se kana na hapchulok tu ni ba and hapchulok to neba. Primase have an enzyme in hapchulok to neba. With the help of the enzyme, primase. RNA primer na pagkansida kado kita masagi kaila DNA polymerase la aga kado ni amo si sungya leap하니. leap하니까 then 그다음에다 시중 그다음에 RNA primer 아무나 아무 leap하니. 근데 좀더 RNA primer 아무나 아무 leap하니. 시부가 이거게다 시기 맞다 그다음에다 मतलब form तो है DNA standard complete हो बस है सिगी standard complete हो देखो सिगी standard मतलब सारे fragment है ना form तो आ रही कि मतलब तो RNA primer से DNA standard है ना देखो RNA primer से लोग ने remove तो हुए थे तो लोग ने remove तो हुए नहीं RNA primer से मतलब सिगी site से वो कोई कोई दा ओकाजाकी fragments हो सिगी RNA primer is synthesized in the presence of the enzyme primase, then this part is known as, this fragment is known as Okazaki. These fragments are known as Okazaki fragments. Okazaki fragment has a name. Then in presence of the enzyme, the DNA-dependent DNA polymerase, many nucleotides join with one another to form strand and form a protection. अगर सीकी समझने सिने कनाने समझने नो हाई मतलब DNA dependent join together सीकी कनाने समझने like ये सब इंटरनेट समझने The Okazaki fragments are then joined together to form a newly strand by an enzyme called DNA ligase. DNA ligase ने समझने नहीं. अगर सीकी बापाज़ ने कट तब फाउ इधर ना. मतलब leading strand इधर जा करने ना मारी थूला जा करने बा. Whereas लगी leading strand then this newly strand is called leading strand के दो बात. सही मारी तब ना गारे ले जो ना हरी मारी मतलब fragment ने form तो उधर नहीं ना. दो के दा मारी leading strand ना कोई. Tapoi legging strand say Okazaki fragments in a form together. Then if the wrong base is introduced in the newly strand DNA polymerase can do the proofreading. Proofreading is who DNA ka gumba sagi wrong base or ba sam nagi edin lakada da thamin laksake laksalaba adu tadi soy da soy tadi ke the proofreading na mu sky the soy da ke da mu check to ne so proofreading ko sila ko Kena sige picture din na in eukaryotes ta tao. Eukaryotes di magi enzyme je different way ni ko. Eukaryotes di magi enzyme ga different tao ni class 2 ke syllabus ta di yaw de eukaryotes tang tamani. Ada mali hekari topoisomerase ko topoisomerase na sagi. Ke topoisomerase tao di sina lagi ke tension na tadi tension na masih kai matam de ka dau ni. Tension na create tao ni ma to ti de. Si tension ji kana na release tao bu na ha matam da. Topoisomerase release tao ni. Then SSD proteins, SSD proteins is single stranded binding proteins na kaya dawi sa secondary structure form to doon hindi so din na kick mo kot daw doon na ko sinang prevent to yan ada DNA polymerase delta sinang sila mali correct different no ko enzyme sing ada primase do chama na re sinin ko exam na dum DNA replication fork si u kaina dum hang mo ni ko replication fork si dum important is an important one ada kari direction da replicate to hata di 5 prime to 3 prime kari direction da replicate to we 5 prime to 3 prime da habdi mai ke template ki strand di template ki strand na kai or ni 3 prime to 5 prime ani since no concept in 3 prime to 5 prime aduga synthesize to but leading strand na 5 prime to 3 prime jala Feeling what? So termination ki process na first initiation tau be initiation ni kanana initiate tau be ni RNA primerase replication fork to fork same maga replication la replication fork form tau ni RNA primer la the initiation then after the polymerization tau then matung sida termination. Why last termination? Had the replicate of the lobe array So termination and replication occurs in different ways in different organisms. In E. coli-like organisms, chromosomes are circular, and this happens when the two replication forks between the two terminals is made. The chromosomes in the E. coli, for E. coli have the prokaryotic cell, particularly chromosomes are circular web. Circular web, come and leave terminate the From side replication fork ani separate replication circular orabanina sineva DNA has circular standard DNA has circular. This circular is a replication fork anisha. But the two replication forks in mid tower the replication fork and the side replication fork on the replication forks and replication fork. Is a kind of size to the joint tower ganda. Can you get a replication fork and it's a joint tower ganda. Terminate our hiring name. Then equalize complete replication within 18 minutes. Why the catch on ye? The replication time two processing commodity 18 minutes. That is 2,000 base pair per second. Ada mana sumbation na 2,000 base pair maximum mga per second. Second naman da, how many base pairs? 2,000 base pairs. In eukaryotes, the replication of DNA takes place at AS phase. AS phase adi na cell cycle the AS phase ata the synthesis phase ada. M phase, AS phase tadra. Ada replication ni kada tawgi AS phase before mitosis or meiosis tau dina the AS phase the DNA sa replicate the whole DNA. DNA si replicate ang tau dina ko. Ano hai si? Which result sisini ko. In the failure in cell division after DNA replication results in polyploidy. Sagi mitosis tau ganda cell ano nila chap mana DNA pigi do siya. DNA chap mana ba gen thukini do. Gen thukpa ngam dra ba fail or ba adu eta di polyploidy. Nilaka niya. Consider DNA replication or DNA replication for Watson and Crick na proposed to be. Then nature is semi-contributive. Then how do you know RNA give world no. RNA was the first genetic material. There are evidences to prove that essential life process such as metabolism, translation, splicing, etc. have evolved around RNA. RNA is involved in DNA is a genetic material in hybrid. DNA is a genetic material. DNA is a genetic material. DNA is a stable stable. RNA is highly reactive name. Highly reactive name. Highly reactive name. So RNA we can get something. Mani life process and for example metabolism, translation about protein sumba, protein synthesize to RNA the kai sema no. Protein synthesize to then splicing sigumba RNA the luin. There are some important biochemical reactions in living systems that are catalyzed by RNA catalyst and not by protein enzyme. RNA na enzyme certain enzymes so very important enzymes RNA is no catalyst not by enzyme. Then DNA has evolved from RNA with chemical modification that makes it more stable because RNA being a catalyst was reactive and hence unstable. RNA has evolved from RNA DNA is lack of DNA is RNA is DNA is simple. Have evolved evolved kay all ko na DNA. Habadi RNA is unstable or banina. May stable form siya po yan. Stable form na kay all DNA siya po din. DNA is na mo kada genetic material o ado RNA is na first genetic material or bani ado mo hojikti evolution ni matung ina DNA is na genetic material o The RNA mo kal mayam lay ko. RNA is 3 types of RNA lay. Kay no mRNA ba man tRNA ba man rRNA ba man mRNA is na messenger RNA, tRNA is na transfer RNA, then rRNA is na ribosomal RNA. Then mRNA is na kado no. It's exit template for transcription. Transcription na kay hagi. DNA se mo sina kay neba. DNA to DNA. This is a replication. DNAdagi protein sembra. DNAdagi protein synthesize tobuni ko. Kasagi wari dina evolution wari ni synthesize to habati RNA na hana first form hari life infused water na water dagi amade amino acids ko kamyam se le madagi RNA form to re RNA dagi kari olai no DNA olai ni DNA sina mo organism DNA ga RNA ga like ta organism sima panera so DNA is evolved from RNA habane DNA form to jina kana nan form to wi RNA nan form to habane but koina sumnagi kasagi transcription RNA Procedural Dogma matungina DNA dagi kari sema ko dune ba DNA dagi RNA se genetic RNA se synthesize to sibu kai ko wi transcription. This is known as transcription. Transcription ko then RNA dagi amuk kari form tor ko dino kari synthesize tor ko dino hamatum na protein sisina kai ko wi translation. It provides a template for transcription. Asagi mRNA sina transcription form to boda transcription ni template oina ko template oina man mo de shama ko de template ata di mo de shama ko de a template oina ekto but it's tRNA is a transfer RNA which brings amino acid and reads the genetic code ado sagi kari de mo da sagi tRNA sina Magi chana ba amino acid pura lakh to ni mo. Then RNA, sina ribosomal RNA is structural and catalytic role in the translation. Translation da madi catalysis so ina catalytic role in ekto. All the three RNA needed for synthesized protein in the cell. And three RNA ahum siya protein sumbada protein anadine. So messenger RNA my structure is in a habit or this is mRNA then ribosomal RNA then madi larger subunit anin sagani this is a larger subunit then a smaller subunit and this is a ribosomal RNA then mari subunit anin sagani larger subunit no smaller subunit. Transfer RNA sida mga on the man sida pula to sida amino acids pula ni amino acid pula ni sida amino acid se amino acid mayam lai ko sina alanin adi valin ni kakaman si amino acid mo pula to ni 20 amino acids na ta 20 amino acids kayo pula ni sila transfer RNA may structure sila mRNA ribosomal RNA transfer RNA that's transcription. Transcription it is the process of copying genetic material from one strand of the DNA into the DNA into RNA. Transcription badi kay ni ba DNA deki RNA simu transcription. The principle of complementary governs the process of transcription as the adenosine now forms based on uracil instead of thymine. DNA application kay doba sagi idindo kanaga base pair form to ba. Thymine nga form to na dugo RNA dadi kawai to gida sida si kay naman adenosine do. Adenosine si kay naman base pair with uracil. Thymine is replaced by uracil in RNA. In transcription, only a small segment of DNA is duplicated and one of the strands is copied into the RNA, but the strands are not copied. At DNA application, what the strands in the template or in the eighth of the parent strand animal template in the eighth of the other template, newly synthesized DNA or a continuum. And the parent stand the meow name. Whereas in transcription, both the strands are copied over. Both the strands are not copied, only one strand is copied never. I'm a strand, I'm going to copy over it. If both the strands code for RNA, two different RNA molecules and two different proteins would be formed. Both the strands to form both the strands to template in the X of the two different RNA molecules and two different proteins and different protein form RNA. This will be very complicated. Complicated or doing Then since two RNA predicts to be complementary to each other, they would form a double-stranded RNA without translation, making the process of transcription futile. Aduna kaigumba sige two RNA na system puthulakpa taraba samna haga sige man hai se DNA ki strand DNA ki RNA same hai bana to si DNA sile DNA ki template am khatang lo hai ba ama di lo tip for example sige 5 prime to 3 prime okay. Pada template cha kadogi da template am khatang lo ani ठीक 3 prime to 5 prime 5 prime to 3 prime 3 prime to 5 prime is a template and act as and is referred to as a template strand. Template strand lo to si karam mane ba DNA strand gi 3 prime to 5 primes oiba pot to sisina template na lo to dim. Aduga 5 prime to 3 prime oiba sina kai kong ki da madi RNA gi sequence manaduni na it is known as coding strand. Coding strand. Habidi sigi pot cha sidi ke ba DNA strand ka template in lo ni ba. Sigi 3 prime to 5 prime in lo ni sidi sumuni RNA sidi so ko ni sidi RNA si me to sidi me to DNA matattagi RNA shin ni si RNA si karam ba ora mani di 3 prime to 5 prime ora mani 3 prime to 5 prime ora mani Shani ko aduna animak kai kumbal complement each other animak sigi strand de same ba sidi RNA same ba sidi same de strand ama tang sigi strand sidi gi DNA same ba Karamba strand 3 prime to 5 prime takpa strand idagi RNA same dune adu sige DNA strand 5 prime to 3 prime DNA strand di idika dowi RNA I template oide RNA template oide adu ne kai goma siso RNA template ora ba sidi sima so RNA masime na khalo adu itardi ani se samjana gi ba yai complement ora ba RNA sidi ka single strand ko double strand nate RNA single strand ko damni double strand nate the single strand oi the double strand oi ta di the transcription will be transcription process this for recorded only one strand acts as a template during the transcription process called during the transcription Then the transcription unit in DNA is defined as 3 regions in the DNA which are as follows. The promoter structural gene terminator promoter is in a how DNA transcription promoter then structural gene the structural gene coding for particular is flanked by the promoter see structural side Howram dagi sim size of as structural gene coding for a particular protein is flanked by the promoter and terminator. Then terminator is the region side transcription loire na hangala ko mapama sisina terminator. The two strands of DNA opposite polarity and the DNA dependent RNA polymerase also catalyze the polymerization in only one direction, that is 5 prime to 3 prime. Sige, sila na kay hani amano na DNA replication na kay haba DNA dependent DNA polymerase haba. Si sila RNA polymerase na lakapit. Ko RNA ki strand sumo kado siya. Kay na sumo kado na RNA polymerase sumo kado niya. Tadi direction na chatli 5 prime to 3 prime direction na chat. The strand is a polarity 3 prime to 5 prime. 3 prime to 5 prime is a template and is referred to as template strand. 3 prime to 5 prime na chat pa sisina kay daw ra is a template. Amma sina 5 prime to 3 prime doon kay daw imadi RNA di strand di direction ko chama na ni adu ko kay doon mas kay na ko ba coding strand na ko. Kay at the sequence same as RNA. RNA do ko chama na hadi may strand sequence man na to ni na 캐나다지 그거. 캐나다지 자기 타이밍이 오다. 리아스 신경락도니. 시든 캐나다가 도이나 그도게도 마시 RNA가 만나게나 거. RNA strand가 만나게나. This strand is called as coding strand. The promoter and the terminator link the structural gene transcription unit. The promoter is located towards the 5' end of the structural gene. The structural gene the 5' end microbiome the lady. To 5' end microbiome the lady to shida. Structural gene. Promoter can be defined as a DNA sequence that establishes a binding site for RNA polymerase. It is located in the 5' end. Magical delay 5' end is the located all the promoters. Upstream with the matone by the lady name 5' end. Of the structural gene. Structural gene gave 5' end is the lay. Structural in structural gene, right? And they also defined as a template and coding strands. By switching expression with terminator, the definition of coding and template strands could be reversed. Other than among see my passing and pay or pay at the hammer of the reverse of the adding hands, see my passing on high see among terminator or terminator the sequence or a boya and have it. Position is for structural gene. The structural gene coding for a particular protein is playing by the promoter in terminator. The terminator has a replication transcription already having a partner. Terminator and high. Okay, nasi this is about Thursday. Next class as in the okay, have a great day. Stay tuned.